Okay, potassium channels with the biophysical properties of what was later identified as Kv1.3 were first described back in 1984 by Michael Hale, Tom DeCourcy, and George Chandy at UC Irvine. This, of course, raised the question, why do T cells need voltage-gated potassium channels? By now we know that immune cells express a whole ensemble, a veritable orchestra of ion channels. And we know that the two potassium channels regulate membrane potential and calcium signaling. Oops. But why bother to work on these channels? Cyclosporin has been invented. Turns out the two channels are differentially expressed in different T cell subsets, suggesting that you could pursue them as targets for more specific immunomodulation. Let me introduce the players here. KC3.1 is a voltage-independent calcium-activated potassium channel. The structure was published by the McKinnon Lab in 2018. They managed to capture the channel in both the closed and the open state with the associated calmodulin. And like Stefan alluded to, we for a long time had a small molecule blocker for this channel, TREM34, which we could use as a tool compound. Kv1.3 is a voltage-gated potassium channel. The structure was recently solved by George Chandy and Shashi Bhusha, and at the same time by Kenton Schwartz at NIH. Kv1.3 is blocked by SHK peptides at picomolar concentrations, and by our small molecule PAB1 at nanomolar concentrations. Kv1.3 blockers are effective in autoimmune diseases. T cells expressing high levels of Kv1.3 have been described in MS, type 1 diabetes, rheumatoid arthritis, and psoriasis. SHK186, oops, here it is, SHK186, which is also called dalaxatide, is effective in animal models of MS and arthritis. It finished IND-enabling toxicity studies, and it was demonstrated to reduce psoriasis severity in a small phase 1b study in psoriasis patients. So why are we now proposing Kv1.3 blockers for neuroinflammation? Kv1.3 blockers are mild immunosuppressants, which is exactly what you want for stroke and Alzheimer's disease. And it was actually described more than 20 years ago by Leon Schlichter's group that Kv1.3 is expressed in cultured microglia and involved in oxidative burst and microglia-mediated neuronal killing. And in our own work, we had seen Kv1.3 staining on cells with a glia morphology in MS brain. These are certainly not T cells. And we had seen Kv1.3 staining on microglia surrounding amyloid plaques. Microglia are interesting because they contribute to the pathology of many neurological diseases. Various stimuli like aging, A-beta, alpha-synuclein, ischemia, hypoxia activate microglia and turn them from cells that are predominantly anti-inflammatory and phagocytotic into cells that produce inflammatory mediators and that can damage or even kill neurons. What does Kv1.3 do in microglia? It seems to be the same as in T cells. It regulates membrane potential and calcium signaling. If Kv1.3 is blocked, microglia can no longer resist depolarizing current injections, and they flux less calcium through P2X4 and through crack channels. Downstream, Kv1.3 inhibition prevents nuclear translocation of NF-kappa B. If you activate cultured microglia with a classic pro-inflammatory stimulus LPS, you see increased Kv1.3 expression, And you can prevent cytokine production and NO production with Kv1.3 inhibitors. But can you really trust cultured microglia or antibodies? We all know ion channel antibodies are terrible. Can we record currents from the real thing? To do this, we developed a technique to acutely isolate microglia out of rodent brain with magnetic beads. And if you do this fast and cold, they are viable enough for a few hours of electrophysiology. Employing this technique, we found that after ICV injection of LPS, there's increased Kv1.3 expression on microglia from wild-type mice, but not from Kv1.3 knockouts. There are no sex differences in Kv1.3 expression in microglia, not in culture here after stimulation with LPS, and not in vivo when we acutely isolate microglia after ischemic stroke. And this is really different from P2X4, where there are significant sex differences in microglial expression. So for Kv1.3, we don't see any differences between males and females. The model we're using here is ischemic stroke. What has currently become the standard of care for stroke is what is called endovascular therapy, which basically means that if you're lucky enough to live close to a center that does this, a surgeon will advance a stent retriever into the vessel and pull out the blood clot. A really good model for this, and also for the reperfusion that comes with this procedure, is middle cerebral artery occlusion, which my surgeon, Dr. Chen, who runs the microsurgical core at UC Davis, really has down to a T. If you do MCAO, you see increased Kv1.3 expression that increases together with microglia activation, day 2, day 5, day 8. This Kv1.3 expression is localized to CD68 positive, INOS positive microglia. So these are inflammatory cells. If we acutely isolate microglia after ischemic stroke, we see the same increase in Kv1.3 expression at the current level that we had seen by immunohistochemistry. We next treated mice after MCAO with our Kv1.3 blocker PAB1, and we see a reduction in infarct area, an improvement in neurological deficit, and less interferon gamma and IL-1 beta in the infarcted hemisphere. Interestingly, Kv1.3 inhibition does not affect phagocytosis of neuronal debris, which is actually important because this means the infarct area gets cleaned up and doesn't stay swollen like the infarct does in TREM2 knockout mice. What I've shown you so far was done in young male animals. And of course, the question in the stroke field is always, does your treatment also work in females and in aged animals? So we did MCAO in young and aged male and female, wild type and Kv1.3 knockouts. And since we now had a lot of mice and a lot of groups, we switched from immunohistochemistry to T2-weighted MRI for analyzing infarction. In keeping with literature, we found that young females had smaller infarcts than young males. But then when we went out to 20-month-old mice, we found that there no longer were any differences between males and females. And 20 months roughly corresponds to 65 years in humans. Kv1.3 deletion provided benefit in young males, but it did not provide significant benefit in young females. But then when we looked at aged animals, we saw that Kv1.3 deletion benefited both males and females. We then looked at drug treatment in females. PAB1 was started 12 hours after reperfusion. And we see a benefit in both young females, but then again, especially in aged females. If we look at the infarcts in aged females, we see that Kv1.3 expression is, of course, on activated microglia here, but we also see staining on T cells. And then when we evaluated microglia activation and T cell infiltration, we find that both drug treatment and genetic deletion reduce microglia activation and reduce T cell infiltration. Here's some target validation in humans. This is Kv1.3 staining in a microinfarct found post-mortem. This is a needle biopsy from an ischemic stroke, so the patient was still alive, taken 7 days after the stroke. And you see Kv1.3 staining on iNOS positive microglia. And here you see an Alzheimer brain biopsy, Kv1.3 staining on microglia surrounding an amyloid plaque. So we wanted to see, do Kv1.3 blockers work in Alzheimer mouse models? So here we have APP-PSI mice, which get a PAP1 medicated diet for 5 months. And you see a reduction in microglia activation, in inflammatory marker expression, an improvement in cognitive deficits, and a recovery of hippocampal LTP. Srikant Rangarayu, our collaborator at the University of Emory, used a fluorescent SHK as a probe to look at microglia in the brain of 5XFAD mice and in post-mortem human brain. And he found that the Kv1.3 expressing cells are CD11b positive, CD11c positive, CD45 positive, suggesting that these are microglia. He confirmed this by transcriptomic analysis. So Kv1.3 is co-expressed with canonical microglial genes and with disease-associated pro-inflammatory genes, so TSPO and IL-1 beta, for example. If you do pathway analysis, which I really don't understand, Kv1.3 expression is downstream of the TREM2 immune checkpoint in a beta-driven microglia activation. But then mechanistically, at the end of the day, this all comes back to membrane depolarization. Kv1.3 inhibitors depolarize microglia and prevent A beta-stimulated activation. We also collaborated with Anumanta Kantasamy at the University of Iowa and looked at Parkinson's disease and found that Kv1.3 inhibitors have beneficial effects in 3 different models of Parkinson's disease. So in summary, we would like to propose Kv1.3 inhibitors as potential targets for inhibiting pro-inflammatory microglia functions in ischemic stroke and other neurological diseases. But there was a second channel. What about KCS3.1? We also see increased expression of KCS3.1 in microglia after ischemic stroke. Our KCS3.1 blocker, TREM34, as well as genetic deletion of KCS3.1, reduce infarct area in stroke, improve neurological deficit, and reduce microglia activation. Same in Alzheimer's disease. There's increased expression of KCS3.1 in the brains of the 5XFAD mice and in patients with Alzheimer's disease. Which is why the clinicians at the UC Davis Alzheimer's Disease Center kept asking, can we do a clinical trial with a KCS3.1 blocker? Our own compound is of course not suitable for this, but there is Sinicapoc, which is a compound that Icagen had taken into clinical trials for sickle cell disease. And this compound has an IND, and it's binding to the same site as our drug. So my chemist Vikrant synthesized 100 grams of Sinicapoc, and then he looked at brain penetration, which is actually very nice. We tested Sinicapoc in our stroke model and found that it reduces infarction and improves neurological deficit, which is why we're trying to repurpose Cenicapoc for ischemic stroke in collaboration with Jonathan Weinstein at the University of WashU in Seattle. And at Davis, my collaborators Izumi Mezawa and Li Wei Jin gave Cenicapoc medicated diet to the 5X FED mice. And we see a reduction in inflammatory marker expression, a reduction in A beta deposition, and again, an improvement in hippocampal LTP. And actually, based on this data, the UC Davis Alzheimer's Disease Center was awarded a clinical trial grant from the Alzheimer's Association to do a phase 2A study with Cenicapoc in mild and prodromal Alzheimer's disease. Unfortunately, this trial was on hold for the last 2 years because of COVID, but we finally started enrolling the first patients. And since we had drug material, UC Davis also provided Cenicapoc tablets to Ulf Simonsen at the University of Aarhus to do a study in COVID-19 associated severe respiratory insufficiency. Unfortunately, like so many COVID trials, this trial really was a bust. There was a reduction in 28-day mortality, but it wasn't statistically significant. And dexamethasone had just come along, so 90% of the patients got dexamethasone before they got Cenicapoc. So there really wasn't anything to analyze here. But let me come back to neuroinflammation. Many of you are now probably wondering what I wonder every day, which channel is a better target, KV13 or KCS3.1? And at this point, I honestly don't know. At some point, we combined our two compounds, PAR1 and TREM34, in MCAO in rats. And we saw again the same 50% reduction in infarct area. There was no synergy, there was no additive effect. And interestingly, Midori Yinari's group tested a crack channel blocker in traumatic brain injury, and again saw a 50% reduction in infarction. I'm now, of course, comparing apples and oranges here, but I would like to throw the hypothesis out there that there maybe is a ceiling effect on how much you can elicit by inhibiting microglial calcium signaling. And with this, I would like to close and acknowledge the people who did this work. First of all, Jay Chen, the man with the magic hands, who did all the surgery. Hainu Yan, a really talented electrophysiologist. Yanjun Ku did the staining. Vikram Singh synthesized Cenicapoc. Ruth Lee, who is here, is our qPCR queen, and she has a poster on the Cenicapoc work here. And you might also want to check out Joshua Nussberg's poster on KCA channel activators. And otherwise, I would like to thank our collaborators at Davis, University of Iowa, WashU, and at Emory. And thank you for your attention. Yeah.
We called it SK.
Yeah.
The question I have, I wish I had those tools that you have now back in my PhD. But my question for you is the very question you asked at the very end, which is which channel is more important? Could it be that it's when the cells are activated that one channel becomes more important than others? So could it be in a disease context that one channel plays a greater role than another? Is there a way for you to explore that?
You know, there's also the memory T cells. So in many situations, especially in aging, I think KV13 might be the better target because the KV13 knockouts age very gracefully. There is a lot of T cells in the brains in the context of aging. But on the other hand, KCS3.1 is not only on immune cells, it's also on the vascular endothelium, it's on dedifferentiated smooth muscle, and in people, Alzheimer's always comes together with atherosclerosis. So I really don't know. You can make a lot of arguments for each channel.
I mean, it's maybe also interesting to comment that microglia is also a very diverse set of cells, right? My colleague Shane Littleow would be here, who is actually a neuroscientist. I didn't mention in my introduction. He would point out that they come in very different flavors, and depends on whether they're inflammatory or more resolving, right? So, and then, do you have any data on what the channel expression pattern is on the various kinds of microglial cells?
So we've looked mostly in the context of more classic inflammation. I always like stroke because it is extreme inflammation. I mean, if you look here at stroke, you don't even need a microscope anymore to see the microglia activation. Typically, I mean, this is now fluorescence, but if you do this with DAB, you just hold the slide against the light and you see the infarct. So this is microglia activation at its best. In Alzheimer's, Parkinson's, we also have a model where we do organophosphate-induced seizures, and then you get inflammation from the seizures. It looks very different, and we've seen situations where the channels actually don't come up with microglia activation. And those microglia need to be classified to really look at what it is. But in AD, in stroke, in Parkinson's, we see both channels come up.
Good morning, Dr. Wulff. Hi. Here.
Hi.
My name is Pedro. I'm a PhD student at Columbia University right now. Wonderful talk. Thanks. I would like to know if you have any, if blocking KV1.3 has any effect on the aftermath of the stroke. So talking about residual gliosis after the effects of the stroke.
Yeah, we need to look at this some more. So far, we've mostly gone out to 8 days. And by 8 days, there isn't much of an astrocyte scar yet. We've started to go out to day 35. And by that time, the microglia activation really comes down, and you get this massive astrocytic scar with also some fibrosis around the infarct, and we really need to start evaluating, do KV13 blockers have an effect on this? What we definitely don't see is that, and this has happened in stroke and especially in EAE, sometimes when you withdraw drugs, the disease comes back with a vengeance. We certainly don't see any type of rebound when we withdraw these blockers. But we really need to start looking at the astrocyte scar.
I was wondering if you have some data related to some long-term studies when you block just one of the key of the potassium channels, because I was thinking if they have similar features in calcium release, in potassium release, and maybe if you just block one chronically, thinking on a future therapy, which I assume is what you're looking for, probably the other, for example, the KCA will take over in the signaling because the other is just not functioning because it has been consistently blocked. So it seems like the strategy that you try to do, blocking both, is the best way. However, it's interesting that you didn't find any synergy. So my question is, do you have any data on long-term administration of these blockers or co-administration to see?
Okay, so we've done these blockers long-term. for the Alzheimer's studies because that was medicated diet for 5 months or for 3 months. We've also in the past done them for transplantation. At some point we did aorta transplantation where you then get chronic transplant rejection. And in that setting, actually this is now interesting and comes back to your question. In the setting of chronic transplant rejection, where you get a lot of smooth muscle cell proliferation and fibrosis, the KCS3.1 blocker was wonderful. The KV1.3 blocker did zip, nada. And even the combination didn't do anything more. So I don't really think the other channel compensates to a large extent.